Thank you very much for the opportunity to come speak to you today. I'm going to talk about the Pediatric Emergency Care Applied Research Network. Was asked to give a bit of an overview on the network and then talk a little bit about one of the studies, just to highlight some of the research that we're doing. Because I knew I was speaking mid-morning, I chose something that had a lot of color to the slides, so hopefully it will keep you focused. And the history of PECARN is we've been a funded network since 2001. We're funded by HRSA through their Emergency Medical Services for Children program. We're the first federally funded pediatric emergency care research network, and our goal is to increase the evidence base for pediatric emergency care around the country and around the world. And some people may not know, Dr. Ron Mayo, sitting right there, was one of, I'm not nearly as slick as the younger generation with slides. I'm sort of just plop people in. But one of our, the founding nodal investigators, so really set the early agenda and really was one of the founders of PECARN. So thank you to Ron for the groundwork that you laid that's helped a lot of investigators and a lot of children around the country. So a network is really important. I'll talk a little bit about why we use a network and why it's important. The incidence for the high impact pediatric emergencies is relatively small, and so at any one site it's difficult to accumulate enough patients to get your outcomes of interest, particularly with rare things like C-spine injuries in children. Also, having a network obviously increases your geographic diversity, makes studies more generalizable, and it also brings in investigators from around the country and lets them have contact with the network. So we get a lot of benefit from being part of this network. And you can see we're now fairly geographically diverse. We've gotten more geographically diverse over the years. Our latest node down in the green is the Southwest node. That was an area where we started off being all north of Mason-Dixon, and now we've expanded down to the south and now into the Southwest. We also, if you look at the pink dots, the CHAMP node is a relatively new node that focuses just on pre-hospital care. So our network is moving a little bit towards pre-hospital care, and pre-hospital care is difficult. Pediatric research, pediatric emergency research is difficult, so combining those is really a challenge. And so we're trying to, by building in a node focused on that, to address some of the pre-hospital issues in children a little bit more effectively. And you can see our node there in, now that our nodal center has moved down to Columbus. We are the Scarlet node. I'm sure Will Moyer approves of that. The Great Lakes Emergency node is what we're involved with. So this is the network structure. I know this is sort of a complicated slide, but every hospital in PECARN has a hospital emergency department affiliate, a HEDA PI. I'm the HEDA PI at University of Michigan, and we all make up the steering committee. And the PECARN steering committee is what takes votes and sets the research agenda for the network. Within the steering committee, there are subcommittees. There's protocol review and development. I'm currently the chair of the Quality Assurance and Safety and Regulatory Committee, Feasibility and Budget. Sherry Goldfarb, who's sitting right in the front, has been an active member of the FAB committee for many years and is a huge asset with budgeting issues. And then grant writing and publication really helps us have a successful and high quality work product at the end of the studies. We also have the Data Coordinating Center out in Utah. Mike Dean, some of you may know, is the PI of that, and of course our federal collaborators as well. I'm not going to walk you through all the way through the PECARN process. I put that up to demonstrate that it is a dense and sometimes challenging process, but at the end we have a lot of success. So investigators come to PECARN from all over, and many, if not most of our study PIs are not PECARN HEDA PIs. They'll come to a node, they'll present a concept, and then that will get approved initially through our federal partners and then go to the steering committee. And there are 3 different levels of periods where the steering committee can input on the concept. So there's the concept where we decide if we want to study a particular topic, if it seems right for the network. And assuming that, that we take a vote and that passes on, then it'll go to the protocol development portion. And protocol will be developed. It will go to the different subcommittees that I showed you. The subcommittees will then look at the protocol, give feedback to the investigators. Oftentimes the protocol will be changed or further developed, come back to to the network, then there'll be a vote, and hopefully as this passes through, then the grant will be developed, and again, another vote, the grant goes out the door, and thus far, we've been really very successful as a network. So this is a little bit of an old slide, so some of the numbers have changed, but you can look as we go from concept to approved concept to protocol, and then when we look at grants submitted, we're at about 80% funding rate, which is really good in this climate. So what it means is that the NIH groups that are funding these studies and looking at it know that if PECARN has signed on to do a project, that there's a very high likelihood of successful enrollment and a high quality study, so they've been willing to partner with us. So some of the topics that we've already looked at, one of our early studies was whether or not steroids are effective for children with bronchiolitis. We've looked at head CTs in kids with mild traumatic brain injury. This is probably our most well-known study as a network, and when I'm working in the PEDS and I get transfer calls from even small community hospitals, they'll tell me on a head injured kid, oh, well, we did the PECARN rule and we did do a CT or we didn't do a CT, but it's nice to have that feedback and know that we're making an impact and we're getting out into the community setting. There's been a decision rule done on abdominal injuries. A different topic that I'll talk a little bit more about later is whether or not we can use RNA technology to figure out which infants have bacterial versus viral illness. We've looked at C-spine injuries in children. We looked at that with a large retrospective study. That was actually my first study within the PECARN network, and we're hoping to move forward with a prospective study soon to try to develop a better C-spine injury in children. The adult providers have a few good rules out there, and we really don't have nearly the evidence base in kids. We're looking, partnering with our NET folks, on the ESET trial, and we've looked in the past at the best benzodiazepine for first-line treatment of status epilepticus. Now we're working with NET on the best second-line therapy. And we've looked at whether or not we can screen for for alcohol problems in teenagers in the emergency department. So this gives you a sense of the variety of things that we've investigated. Again, we're a population-based network. We look at kids in the emergency department. We're not a disease-specific network, unlike some other networks out there. And we think we've had a pretty good impact. This is a little bit of an older slide, but you can see our publications. They've gone up. It's not that 2015 was really that anemic. It was just that was when this slide was done. So we're now at about 100 publications, and then if you add in national presentations, we're well up into the 200 range. And currently what we're doing at University of Michigan and what I'm involved in, again, I mentioned the ESET trial, looking at best second-line therapy for status epilepticus in both children and adults. So for residents or attendings out there, please think about us if you have a patient who's seizing. We're also looking at probiotics for acute gastroenteritis. There's really no good therapy out there for acute gastroenteritis in kids. And although there's not a lot of mortality from this in the United States, the morbidity, lost work, costs spent is tremendous. So even being able to shorten the course of viral gastroenteritis by a few days would have a huge impact on lots of folks around the country. We're involved with the ED STARS study. This is looking to develop a better screening tool for suicide risk in teens. We know that suicide is a major problem, and we don't have a great way to screen kids for it. This has been an exciting study that we're actually, as a network, well ahead of what we are projected to enroll at this point. And so it gives a sense of how successful we've been when we actually have been, we're at about, I think about 106% of targeted enrollment at this point. And then I mentioned RNA biosignatures, the detection of bacterial disease in febrile infants. I'm going to talk a little bit more about that because we have some past and future to talk about. So this is sort of a typical pediatric emergency medicine case where you'll have a 25-day old infant comes in with a fever and non-focal exam. Most of the exams in this age are non-focal. You have parents who are terrified. They've heard about all the terrible things that we're going to do to their kid. The ED's busy, the hospital's full, all these things that are kind of a typical day. So we do all these things to these infants. We do a cath UA, and we'll do blood work, and we'll put in an IV, and we'll do a spinal tap, and we'll admit them for IV antibiotics for a 48-hour, what we call a sepsis rule-out. It's a pretty standard admission in in pediatric emergency medicine. And the reason why we do it all is because we can't tell which 5 to 10% of kids actually need it. So there's a lot of, there are invasive procedures, there's potential for nosocomial infection, there's antibiotic exposure, lots of things that happen because we just can't tell. And currently our gold standard of blood cultures is something that we have to wait 48 hours on. And so we think there might be a better way to detect what pathogen is infecting these kids. So this is sort of a schematic of it, that just because we don't know what type of pathogen is infecting a child, the child's immune system knows, or we think it does. So if it's a bacterial versus viral illness, there'll be a different immune response, and there'll be a different RNA expression profile depending on what the pathogen is. And we think that by detecting this and analyzing it, we may be able to determine whether it's a viral versus a bacterial pathogen, and even get organism specific as we get better at using this platform. So this is the colorful slide. Obviously, I won't talk all the way through this, but what this is, the columns going down are individual patients, and the rows across are individual genes. So on the left, you'll see the healthy controls. Yellow is about average expression. Blue is underexpressed, and red is overexpressed. So even if you just look at it, you can see that there are different patterns amongst bacterial and viral illnesses that you can start to detect. This is from about it's from 299 samples. So this is fairly preliminary work from a study that we finished a few years ago, and we're going to go on and do the second phase of it that's just starting up soon, and hopefully get a fair bit better with this technology. So current results, 299 samples. There are more to be analyzed. The analysis is ongoing. I don't have slides on more recent analysis, but we're hoping to get more robust signatures to really hone this in to determine what can differentiate viral versus bacterial versus co-infections, which are also common and can be a bit of a confounder. We want to narrow the confidence intervals, so if we think it's viral versus bacterial, we can be more confident in that. We want to potentially look long-term to look at our current gold standard. This is often compared to gold standard, which is blood culture. A problem being that, particularly in infants where you're only getting a few drops of blood, we don't really know what the false negative rate is on our gold standard. So there may, long-term, this may hold promise to be actually better than what we're currently using as a gold standard. And again, to define organism-specific biosignatures, so to be able to say not just that it's a bacteria, but maybe it's E. coli, maybe it's group E strep, maybe there's some way to tailor our antibiotic therapy and really all of our therapy to the specific organism. So the next steps for this are determine the stability. We're going to see if these biosignatures are stable over time, whether or not antibiotic use has an effect, whether antipyretics have an effect. And again, the second phase of this study is going to have a much more comprehensive plan to detect particularly viral pathogens. The ability to do that has increased pretty dramatically recently, and so now we're not just going to look at bacterial positive versus bacterial negative, but what is the virus that may be causing the illness. That's currently under IRB review. That's where we are with this. It's been funded, so hopefully we'll be rolling this out in the next few months. So just some final words. PECARN we have a wide range of research. We've been a funded network for 15 years. Our goal is to improve the evidence base of pediatric emergency medicine in children. We've had well over 100 publications and presentations to date. And again, many of the principal investigators are actually from outside the network that come in with great ideas. So we're always looking for new collaborators. If you have an idea that you think would be appropriate for multi-center pediatric research, we would love to hear from you, and please come find me. PECARN.org if you want more information. And I want to thank Drs. Prashant Mahajan over at Detroit Children's, who's the PI on the biosignatures study, and S.J. Sussman at the DCC for helping with some of the slides. And thank you very much.
I just was curious because you were talking about the blood cultures taking 48 hours. What is, or what do you anticipate the turnaround time to be for the mRNA analysis?
So that's a great question, and that's something that probably like most biotechnology, it's moving very quickly. So this currently, we're batching our samples. So this is not, at this point, a real-time test. There's actually another slide that I had to cut due to time where you can look at all of those little genes that are upregulated and downregulated and do what they call a modular analysis. So you glump them together. That's probably not a technical term of glump, but you put them together and you can get, instead of looking at individual data points, which will need to be analyzed, you hone it down to about 20 that are kind of genes that tend to travel together. The goal is to eventually have a platform that can be run in real time over the span of a number of hours. I think the technology is being pushed, and like a lot of these things, what's possible now isn't what's going to be possible in 5 years. So right now, we're more looking at the concept of whether or not there's truly promise in this technology, and I think that but I don't know how long the individual gene chip takes to run at this point. So someone else in the audience may actually know that.
So this is an example of where there's some potential for synergy between what you guys are doing in PECARN and what Scott is doing in the back. I don't know if he's still here in terms of the synergy between looking at the host response to the infection versus the actual pathogen and detecting the pathogen. And he's been doing work for identifying very small amounts of bacteria in the bloodstream and also the genetics of that and antibiotic resistance. And I don't know if, Scott, you want to comment on how access to these samples might be a nice resource for some of the work that you're doing moving forward.
Yeah, certainly. I actually have a few slides. I have a few slides on this that I'll present this afternoon that talks about an assay that may help that and how there'd be actually a ton of synergy, and particularly to answer the question about how long it would take to do these assays.
I think that's why you wanted us to do this whole thing, right? To develop some synergy. So that's great.
Yeah, well, there's a lot of breadth between clinical research and basic science research in the department. I have a question for you. So you didn't come right out of residency and start doing research, is my memory. Can you just talk for like a little bit about how you got to this place? Because you're having a huge impact with research nationally. It wasn't a path you started on. If you could just take a moment.
So since pediatric emergency medicine, I did a fairly typical career path for that, which is a pediatric residency and then a PZM fellowship down in Atlanta, where we did do some research training, and so got a little bit more training than I had certainly coming out of residency. That being said, I came to University of Michigan interested in research, not really knowing which direction I was going to go in, and got involved with the PECARN network at the time. Ron was the nodal PI. Rachel Stanley was the HEDA PI, which is what I am now. And we often bring in other investigators and other site PIs to try to help out and round out the PECARN site. So currently, for example, Allison Cater, who's right there, is going to be the site PI for biosignatures 2. I was brought on to see if I wanted to help with the C-spine study, which was a tremendously big and somewhat unwieldy retrospective review looking at cervical spine injuries in children, and spent many, many months plugging away at that. And I think that I think with more perseverance than brilliance, sort of earned my spot at the table. It was a lot of work, and then as Ron transitioned out and Rachel moved into the role of nodal PI, got involved. I became the HEDA PI and sort of moved up, and since then have been involved with the network and gotten to work with a lot of, there's great mentorship. You get to develop mentors from all over the country, and I think that's been really helpful in my career. And it's great, even though being part of a network, you're not always the one necessarily doing all the components, and you're not always the one it's not always your project that you get to control all parts of. On the other hand, being able to say that I'm able to contribute to kind of getting some answers to some of the big pictures in our field has really been a great opportunity. So, thanks.